Okay, thank you very much, Jean. Thank you very much for the opportunity to speak to you today. So my name is Matt Campbell. I'm based in Trinity College in Dublin, and I run a research lab that's really heavily focused on trying to understand the blood-brain barrier and the role of the blood-brain barrier in both health and disease states. So what I'll try to do today is give you an overview of the blood-brain barrier, what it is. It's a term that a lot of people mightn't have heard of. And it also is a term as well that sometimes there's confusion as to what the blood-brain barrier is. So I'll do my best to try to teach you what the barrier is and how it can become dysfunctional across a whole range of neurological conditions. And then what I'll do is I'll tell you the story about how I actually came to be involved in AHC research. I mean, up to 18 months ago, I'd never heard of this condition before. So I'm by no means an expert. I'm an expert at the blood-brain barrier, but I'm no means an expert on this disease. And it's a real privilege to actually be in a room filled with people who are experts. And by and large, the experts of this disease are the families. So a bit of background, this is Trinity College in Dublin. So you now have an excuse to come and visit Ireland. So if you're ever in Ireland in Dublin, swing by Trinity College, very old university. It's nearly 500 years old. It was founded by Queen Elizabeth I in 1592. So lots and lots of history, and it's a beautiful campus as well. This is our famous library, the Long Room, built in the 1700s. Again, you walk into this room, you can smell the old books. So you know, these pictures don't really do justice to the magnitude of this room and what it is. It's so impressive that George Lucas actually copied this room in one of the episodes of Star Wars. And I think Trinity threatened to sue George Lucas. They would have got lots of money for research. This is my department. So at the very, very back, the campus is in the city centre of Dublin. And up there on the third floor is where I live. So my lab occupies the entire third floor. And as I mentioned, we really heavily focus on trying to understand the role of the blood-brain barrier and the blood-retina barrier in health and disease. And I really do hope that the stories that I'll tell you about, various ongoing projects in my lab before I talk about the AHC and the gene that we've discovered that implicates the blood-brain barrier in AHC. I hope it'll give you an appreciation of how important this barrier is and how really, any neurological condition at some stage will involve blood-brain barrier disruption. So what is the blood-brain barrier? So open up any medical textbook and look at the cerebrovasculature, the blood vessels of the human brain, and you'll get images like this. Okay, so put your hand on your neck, you'll feel a pulse on either side. That's the carotid artery feeding oxygenated blood up into the brain. Okay, in humans, so unlike smaller mammals, 25%, up to 25% of the daily energy intake in our body. Okay, so look at the calories at the back of the sandwich that you have for your lunch. 25% of the energy within that sandwich is going to make your brain function. Okay, which is pretty crazy if you think about it. The size of the human brain compared to the size of the body, that it's a small enough organ compared to the overall size of the body, but it's consuming a quarter of our daily energy intake. And the reason for that is, unlike other organs, the brain is not able to store glucose in any way. Okay, so the brain needs a constant supply of glucose, and it gets that from the diet. Okay, now that glucose needs to get across the blood vessels, and that's where the blood-brain barrier comes in. So talking about the human brain, we've got 100 billion brain cells make up an adult human brain. And we estimate that no neuron, so no brain cell, is ever more than about 10 microns away from a capillary. What that means essentially is that of those 100 billion brain cells, almost every single brain cell has its own little capillary. Okay, so if you think about that in the context of neurodegenerative diseases, epilepsy, if a neuron decides to get sick and starts to get dysfunctional, eventually the blood vessel, the little capillary beside it, will become dysfunctional as well. What we sometimes think about in the lab is we think about that in the other way around. So what happens if a lot of these diseases could primarily be cerebrovascular diseases where the blood vessel becomes dysfunctional and then drives the dysfunction of the neuron. Okay, so I want you to start thinking about AHC in that context. Okay, looking at the cerebrovascular nature of this disease. The capillary network is so vast, so 100 billion brain cells would mean a lot of capillaries. It's so vast that if you take out all the capillaries and line them up side by side, they'd stretch from Dublin to Paris, 600 kilometers. The surface area of the blood vessels in the human brain is the surface area of a tennis court. So really these medical textbook images don't do justice to the sheer density of blood vessels in the human brain. And we need to look at these vascular casts of the human brain. So what this is actually is it's a polymer that's been perfused into a cadaver. Okay, the brain is then removed from the skull, and the brain material is digested away, and what you're left with is a vascular cast. So it gives you a real appreciation of the sheer density of blood vessels, and remembering this idea, this mantra, that every single brain cell, every neuron has its own capillary. So a cartoon schematic of what these capillaries look like. There's three main cells that make up the so-called blood-brain barrier, okay? The first cell type is the endothelial cell. So when you hear that term endothelial cell, it's a cell that lines a blood vessel, okay? So the endothelial cells line the blood vessels. A second cell is called a pericyte. A pericyte is like it's equivalent to a smooth muscle cell or a fibroblast. It helps with contractility, so it helps with the pulsing of the capillaries. So the pericyte is the second cell, and the astrocyte is the third cell. So you get these foot processes of the astrocyte that wrap around the capillary. So all these three cells work together to form what's called the blood-brain barrier, okay? And that term, the blood-brain barrier, is a concept, okay? It's simply the properties that endothelial cells in the brain have compared to endothelial cells have in other parts of the body, okay? These cells tightly regulate what gets into and out of the brain on a daily basis. And we know from our research and research in other labs that these endothelial cells can become dysfunctional across a whole range of neurological conditions and actually drive the clinical phenotypes that we see. My lab is really, really heavily focused on what are called tight junctions. So remember these endothelial cells, at the point of contact of two endothelial cells that are beside each other, you have a thing called a tight junction. And essentially what the tight junction is, it's a network of maybe 20 to 30 interacting proteins that essentially seal off the gaps between endothelial cells. And the brain endothelial cells have tight junctions unlike any other tight junctions in any other part of the body. They really regulate what gets into and out of the brain between the cells, or what's called the paracellular pathway. And that becomes important for AHC, and I'll talk about that later on in the talk. The most important gene, and subsequently the most important protein, so the protein product of the gene at the blood-brain barrier, is a gene called Claudin-5. Okay, it's a small protein, 23 kilodaltons. So when you talk about kilodaltons, so that's 23,000 daltons. A dalton is probably about the size of one atom. Okay, so Claudin-5 as a molecule, as a protein, is about 23,000 times the size of one atom. Okay, so it's a relatively small protein. It's what we call a 4-pass transmembrane protein. So us scientists, we're very, very unoriginal. So you say what you see. It's called a 4-pass transmembrane protein because it passes the membrane 4 times. It has these loops that come out of the endothelial cell and interact with another Claudin-5 molecule on an adjacent cell, and they essentially seal off this paracellular pathway, okay? So nothing really can get between two endothelial cells, the blood-brain barrier, except maybe for the smallest of ions. Obviously, we saw the talk earlier on from Professor George about animal models. So we do make a lot of animal models in the lab, and I hope I can convince you that our model that we have is very, very similar to the model of AHC. This is a Claudin-5 knockout mouse, okay? So this is a mouse that completely lacks the Claudin-5 gene. And what we can do with these animals is we can inject what we call tracer molecules, molecules that we know won't get into the brain if you inject them into a vein. And what you can see here in this mouse, this is a wild type or a normal mouse. A dye has been injected into the mouse, and the dye circulates throughout the body, but it goes everywhere in the body except for the brain and spinal cord. And that's because of the blood-brain barrier. The endothelial cells that form that barrier are preventing the entry of this small molecule from getting into the brain and depositing in the brain. So these are classic blood-brain barrier experiments. You inject animals with low molecular weight dyes, and you see whether the dyes get into the brain or they don't. But look what happens when you knock out Claudin-5. The dye goes everywhere in the brain, okay? And this mutation or this knockout is embryonic lethal. So you need Claudin-5 to live, okay? So these mice have no Claudin-5, it's embryonic lethal. The animals get born, but they die within about 10 hours, okay? So when that happens, it's called a dosage-sensitive gene. Claudin-5 is a dosage-sensitive gene. Its levels are really, really important for maintaining health. So the first story I'm going to tell you about in my lab, I'm going to tell you 3 stories. One is on epilepsy. The second one is on COVID, which we don't want to hear about, but there's some interesting data that I want to talk about with COVID. And then the third one is on AHC. So the first story is on epilepsy, and more specifically temporal lobe epilepsy, which is one of the most common forms of epilepsy in adults. Epilepsy is interesting as a condition in that there are drugs, really, really effective drugs that can treat and manage seizure activity in patients who have epilepsy. In Ireland, we've got about 5 million people in Ireland. We have about 37,000 people in the country who have temporal lobe epilepsy, okay? Now, while the drugs are amazing and they work very, very well in some patients, about a third of patients become or are never responsive to these drugs, these anti-seizure medications. And that amounts to between 11 and 12,000 people in Ireland. And it's, you know, the quality of life of these individuals is really, really diminished, okay? They've either started the medication and it stops working, or else they've never been responsive to the medication. There are lots of genes that are associated with rare forms of epilepsy, but in the vast majority of cases of temporal lobe epilepsy, it's sporadic, so we don't know the genetic cause of it. So around 5 years ago, we started a project in Ireland with Professor Colin Doherty, who's the clinical lead in our country for epilepsy. And what we were doing basically is there are some patients in the world who have temporal lobe epilepsy and are refractive to treatment, that their seizure activity is so severe that it sounds barbaric, but they're worked up for neurosurgery. The neurosurgeons will go in and take out the entire temporal lobe to try to stop the seizures. And what we did was we decided to look at the integrity of their blood-brain barrier before the surgery, and then in the weeks and months after the surgery. We wanted to get a good feel for what is the role of the blood-brain barrier in this condition, epilepsy. What we also did was we had a unique opportunity that we could actually work with patients, do clinical research, pre-surgery and post-surgery, but also the material that was resected or taken out of the brain, we could bring it back to our lab and actually look under a microscope and see what did the blood-brain barrier look like in these patients before, in the context of temporal lobe epilepsy. So here's the first data set. So this is unpublished work. Aspects of this work are published already. They were published last year. But what you're looking at here is an MRI scan of a young man who is having more than one seizure per week. His duration of epilepsy was for 8 years, okay? And again, uncontrolled. So he had left temporal lobe epilepsy. This is left temporal lobe here. Obviously, the radiological images are back to front. So his seizures were emanating from his left temporal lobe, okay? But what we did while he was lying in a scanner, he had a cannula, a drip in his hand, and we were able to inject a dye called gadolinium, okay? We know the exact molecular weight of gadolinium, and we know that it doesn't cross the blood-brain barrier, okay? What you're looking at here in pseudocolor, these red signals, anywhere where you see red is areas where the gadolinium that's been injected into his vein has extravasated or diffused out of the blood and deposited in the brain, okay? So even though his epilepsy was manifesting and emanating from his left temporal lobe, there was blood-brain barrier disruption throughout his brain, okay? So we did this scan about 3 weeks before he went to Beaumont Hospital in Dublin for neurosurgery. And this was what his scan looked like about 2 months after he had his surgery. Okay, so he had complete resolution of blood-brain barrier disruption, and he's now seizure-free. So you can see the area here, the temporal lobe. There's no temporal lobe anymore, okay, because it's been resected. But this, to our mind, we've done this now in 9 patients in Ireland, and we get the same result with all the patients. Okay, pre-surgery, lots of blood-brain barrier disruption. Post-surgery, the barrier disruption resolves. So to our mind, this is clinical evidence that regulating blood-brain barrier disruption can potentially treat seizures. So as I mentioned, when the temporal lobe came out, obviously lots of it go to the clinical neuropathology for diagnosis, but we get lots of material back into our lab, and we can use our own techniques in the lab to probe and look at the blood-brain barrier under a microscope. And this is what it looks like. So back to Claudin 5. So we know how important Claudin 5 is. You knock it out of a mouse, you get all this permeability of the blood-brain barrier. When you stain normal, non-diseased human donor tissue for Claudin 5, what you see is this beautiful striated pattern of Claudin 5. It's enriched at the tight junctions and stopping the diffusion of any molecules from blood into brain. But look what happens in this young man, this 30-year-old who had temporal lobe epilepsy. The Claudin 5 in red here, it's all in these little patches. It's in these blobs inside the cell, likely in what we term lysosomes. So it's material that's just, it's not moving to the tight junctions. It's being produced as a protein, but it's not localizing to the tight junctions. And what we also see when we stain this tissue for blood-borne components, so components that I know are only produced in the blood, we shouldn't see them in the brain. This is what we see. So immunoglobulin in your blood is full of immunoglobulins. They don't get into your brain if your brain is healthy. Look what happens here. So this is the lumen of the vessel. So this is the actual blood vessel. Anywhere you see red is immunoglobulin that's diffused from the blood and deposited in the brain. And in the same regard, fibrinogen is another blood-borne component that we should only see in the blood and not in the brain. But in this young man, he had lots and lots of fibrinogen leaking out of the blood and deposited in the brain, okay? So this was proof that the blood-brain barrier was disrupted at the molecular level and also at the clinical level, and that resolving blood-brain barrier disruption could actually regulate seizure activity. There's a question though on what is the cause-effect relationship. Okay, so we know epilepsy is an interesting condition in that everyone in this room has capacity to have a seizure. The brain has a threshold beyond which it can't be pushed anymore and you will have a seizure. The threshold will be different for different people, okay? But what we need to understand in this condition is what is the cause-effect relationship? What is the exact role for Claudin-5? Is seizure activity driving decreases in Claudin-5 and seizure activity, or is blood-brain barrier disruption the causative agent driving seizure activity? So two research fellows, postdoctoral research fellows in my lab worked on this project for nearly 4 years to try to understand this in mice, but also in human donor tissue. So what we do in any project in the lab that is translational, has a clinical element to it, we always probe it with multiple animal models. We never rely on just one animal model. The classic model for epilepsy in the world is where we take a glutamate analog called Kainic acid and you inject it straight into the brain. Okay, so direct injection into the brain of a mouse and the mouse gets seizures. What we can do then is we can do the exact same as what we did in the human subjects. We can take the tissue and stain it and look for Claudin-5. This is the contralateral side. So this is the side that wasn't injected, and this is the side that was injected. So here, what you're looking at in green is Claudin-5. Again, the uninjected area, you see this beautiful striated pattern of Claudin-5. But look what happens in the epilepsy model. It's exactly the same pattern of Claudin-5 expression and production that we see in Kainic acid exposed tissue that we saw in the human subjects. And again, what we can do is we can inject these animals with tracers, dyes, that we can then take the brain out and make very thin sections of the brain. And you can clearly see that when Claudin-5 is decreased because of the glutamate analog Kainic acid, you get lots and lots of leakage at the blood-brain barrier. So that's one model that we show that surely enough, when you suppress Claudin-5 in animal models, you can recapitulate what we see in the humans. The second model is a mouse that we made in the lab a number of years ago. I won't go into the genetics of it, but essentially this mouse is what we term an inducible mouse. Okay, so we've introduced genes into the mouse that allow us to decrease Claudin-5 levels in an adult mouse. Remember, Claudin-5 is embryonic lethal. You make a knockout mouse and the animal dies within 10 hours, so you can't really do much work with adult animals. So this mouse allowed us to get the animal up to adulthood, feed the animal a doxycycline, so an antibiotic, that we knew would produce a genetic material called hairpin RNA that would decrease Claudin-5 levels, okay? Now look what happens when you decrease Claudin-5 levels in adults. You get these very, very strange electrophysiological readouts, very, very similar to the data that Professor George showed earlier on. The animals phenocopy, so they look quite similar to the knockout mouse because the animals eventually die. So the longer you suppress Claudin-5, the animals will die. And we have this video. So if you can play that video, Bill, I think it's quite interesting that the model that we make phenocopies the model that Professor George showed earlier on, if it works. So what we see is we see hypermobility and we see seizure activity in these animals. So these are adult animals that have been exposed to doxycycline to bring the Claudin-5 levels down. Not working. We can't see it. It doesn't matter. I can hear it. I can hear the mouse scuttling around. That's the hypermobility. Yeah. So anyway, if anyone wants to see it, just come up to me afterwards and we can show you the video. But it is fascinating that it completely phenocopies. It looks identical to the ATP1A animal. Okay, so I'll have to click that on again. So next slide. Singing an Irish song while we're waiting. Or dance, or river dance. Good? No. You might just come out of it and start it again, but jump onto the next slide maybe. While Bill's getting that organized, just to mention that the cerebral vascular, this idea of the blood-brain barrier disruption across a whole range of diseases. The other diseases that we've seen blood-brain barrier disruption in are dementias. So we see lots and lots of swathes of blood-brain barrier disruption in dementias. And we see it in chronic diseases of the eye as well. The retina is just an external portion of the CNS. comes out of encased bone, and we see lots of blood-retinal barrier disruption across retinal disease as well. Why do they have seizures? So we think it's likely due to a shift in ionic gradients. So if the barrier is size selectively or marginally permeable, leaky, that material from the blood that should be excluded from the brain is just getting access. So you've got things like maybe albumin getting in there, causing astrocytes to become gliotic, to become activated and stiffened, and vice versa. The likelihood is you have ions that should have a higher gradient in the brain leaking from the brain back into the blood. So it's disrupting the neurovascular unit. That's what we think is happening here. Any other questions while we're waiting? Nope. Yes. Yeah. Yeah. Yeah.
When they go to really bad spells, and I wonder if these prolonged spells, they have to do some downregulation secondarily of quantified that's happened during that time, but then recovers. I don't know.
Yeah, no, absolutely. I mean, I'm going to show some data from the AHC study it's called arterial splenomegaly. So it's an MRI technique that looks at blood flow. And during acute phases of hemiplegia, you do see a very, very drastic increase in flow on one side of the brain. Interestingly, with Claudin-5 as well, I know sleep is a very, very big factor in AHC as well. But Claudin-5 is highly regulated by circadian rhythms. So the promoter region, the part of the region of the gene that controls Claudin-5 expression, is highly regulated by circadian rhythms. So that kind of leads us to this idea of, can regulating Claudin-5 prevent seizure activity? And so there's this molecule that Roche Pharma have been developing in Basel in Switzerland called Repsox. And Repsox is really, really potent at being able to regulate Claudin-5. It's a TGF-beta receptor antagonist, and it has very, very strong ability to regulate Claudin-5. So what we did in our lab is using, again, one of the models, the Kainic acid model of epilepsy, where you inject Kainic acid directly into the brain. We were able to show in this model that it actually prevents seizure activity in these animals when the animals are exposed to chronic Repsox. So this was preclinical proof that this molecule regulating the blood-brain barrier could be a means of preventing seizure activity. So this is published in Nature Communications in 2022. And similar to Professor George, we are developing gene therapies in the lab, again, using AAV9 as the serotype. And what we're doing here is we're going direct to source. So we're packaging the Claudin-5 gene into the genome of an AAV and exposing the animals, directly injecting the virus into the brain or systemically injecting the virus to increase Claudin levels to then prevent seizure activities. This is an ongoing study, and we're really excited about this concept. Certainly in temporal lobe epilepsy, the neurosurgeons are willing to go in and take out the temporal lobe. So they are willing also to stereotaxically inject AAV around the region of where the seizure emanates from. So we're really excited about this part of the study. So just to summarize this first story from the lab, that Claudin-5 dysregulation can prime the brain for seizure activity. Okay, so we think this is all about the thresholds, that there are some people who have very, very low levels of Claudin-5 as a threshold in their brain, and it can prime the blood vessels to be very, very sensitive to developing, the brain to developing seizures. Decreased levels of Claudin-5 induce these spontaneous seizures. You can see the video on my mice later on. And we do think that a targeted regulation of Claudin-5 can prevent seizure activity. So we're actively developing therapies for Claudin-5 regulation for temporal lobe epilepsy. So the second story in the lab is about SARS-CoV-2, which we're all absolutely sick of hearing about. And I've no doubt at all that everyone here has had COVID. And also people here will have heard of long COVID, or this lingering symptoms of COVID that you know, people are experiencing. Certainly the friends and family of mine are experiencing this. That's stopped us again, Bill. So we know at the very, very start of the pandemic, back in 2020, there were lots and lots of reports coming out, but they were mostly based on post-mortem donor tissue reports that the blood-brain barrier was being disrupted in SARS-CoV-2. Now, just to caveat this isn't the sickest of patients. These are patients who would have had SARS-CoV-2. They would have been hospitalized, they would have passed away. But what you're looking at here is, in the first images on the top right, is the olfactory bulb of a patient who had SARS-CoV-2, had COVID and passed away. And if, similar to the epilepsy story that I showed you, in green, we've stained tissue here for fibrinogen, a blood marker, which you're seeing it diffuse throughout the brain. So in the olfactory bulb, in very, very severe SARS-CoV-2 infection, there's lots of blood-brain barrier disruption. Other groups subsequently reported later in 2020 and again in 2022 that you get all these microglial cell activation. These resident immune cells in your brain are being activated in SARS-CoV-2. And also these meningeal cytotoxic T cells. There's a strong immune component here where you get systemic immune cells that diffuse into the brain in SARS-CoV-2. So obviously, like all countries around the world, there was a call to action for research. And what we did in April 2020 was we started a study in St. James's Hospital in Dublin, where we got access to a bioresource. So basically all the patients who were coming in unvaccinated in April, May, June in 2020, mostly healthcare workers actually. What we were doing was we were collecting blood samples. We were collecting serum, we were collecting peripheral monocytes, cells in the blood, and we were storing them for future analysis. And what we did was we were able to separate patients into mild. So these were patients who were just coming in because they felt they were so sick they needed to be hospitalized, moderate and severe. So moderate were patients who needed supplemental oxygen, and severe were those patients who were in the intensive care units. And what we did initially was we screened the bloods for all of these pro-inflammatory markers, you know, things like TNF, IL-6. These are the proteins that we know are drivers of inflammation. And they were all up across the board, especially in those patients who had COVID-19. The patients who had moderate or severe were the ones who were coming out with these very, very systemic profile of inflammatory genes. But back to what you were mentioning about the markers, there are markers in blood that can suggest the blood-brain barrier is disrupted. And so we thought, based on all this post-mortem donor tissue analysis, maybe we should look in blood to see if the blood-brain barrier is disrupted in these patients. And one of the main molecules that we can use is a molecule called S100 beta, okay? So if your blood-brain barrier is disrupted, if you had a cerebral infarct, like a stroke or traumatic brain, your S100 will skyrocket. It's been around for years. It's great to tell you when the barrier is broken. It's not great for telling about prognosis, about how well you're going to do, but it's very, very clear sign that your barrier is disrupted. But we saw absolutely nothing, okay? So we were a little bit disappointed. But it turned out that the clinicians who are recruiting these patients and taking the bloods are also asking them about long COVID and about brain fog. And this term brain fog is not too familiar clinically. People who work on concussions in sports therapy, physical therapists will understand the word brain fog. So people who've had a concussive brain injury and are having difficulty, we'll talk about this fogginess that they have, this brain fog. So it's not that it hasn't been heard before, it's just not that well evolved in the medical literature. But they were being asked, did they have brain fog or did they not? And so when we went back retrospectively and looked at the S100 beta levels in the patients who had brain fog, it was increased. When we look at the barrier in these patients who have long COVID with brain fog, will there be any differences? So again, similar to the epilepsy study, I'm working with Colin, we were able to do the MRI brain scans with gadolinium, with this dye again that we inject while the patient is in the scanner. And sure enough, look at this patient here. So this is a patient who has brain fog compared to an individual who has long COVID but doesn't have brain fog. And you see in both temporal lobes, bilateral temporal lobe, really, really, that they've got lots and lots of gadolinium coming out of the blood and depositing in the brain. It's interesting with this, the images I showed you of the patient with temporal lobe epilepsy had blood-brain barrier everywhere in their brain. This is much more specific to neuroanatomic areas. So lots in the frontal lobe, lots in the occipital lobe, lots in the temporal lobe. So in areas of the brain that we know the clinical readout of having damage in those areas is this long-term neurocognitive decrease. So this is all quantified here, I won't go into details, but this was a relatively large study. And we're really, really excited about these results, because if we can figure out what's happening in these brain fog, if the barrier is disrupted, could regulating barrier patients have therapeutic efficacy? Could we find treatment modalities for brain fog associated with long COVID? So we know now that the blood-brain barrier is disrupted in patients with brain fog who have long COVID. Barrier disruption is associated with cognitive decline. So these patients who have brain fog, who have blood-brain barrier disruption, are on cognitive assessment. And we're now looking at clinical ways of stabilizing the barrier using conventional drugs that are available to regulate the blood-brain barrier to see whether it can assist in resolution of cognitive decline. So on to AHC, and a condition that I hadn't heard of up until about 18 months to 2 years ago. So it was interesting that somebody mentioned earlier that obviously you guys remember the first day we heard of the term alternating hemiplegia. Well, I also remember the very first day that I heard of it, and I remember the exact time as well. So it was the 12th of August, 2021 at 3 p.m. in the afternoon. I got an email from this guy, Arnold Munnich, who's a very, very well-known pediatric geneticist in France. He was actually the scientific advisor to Nicolas for almost years, I think. So very, very well known. So when someone like this, you reply pretty quickly. So he had contacted me back in August 2021 to say that he had identified 2 children in France who were unrelated and had mutations in Claudin-5, okay? Mutations in the coding sequence of Claudin-5 that were presenting with symptoms that were uncannily similar to alternating hemiplegia of childhood. They were fitting all of the diagnostic criteria that are laid out in the ICARDI diagnostic hallmarks for alternating hemiplegia of childhood. Now, his group are pure geneticists. They simply just look at genetic information and then they get other people to do the functional work. So he saw my research on Claudin-5 and contacted me and was asking me about the idea of what do I think is happening here with this mutation. Obviously, I had to go and do my own work in the literature, and it came across, I mean, it is incredible. So people like me can actually go and figure out what it is and see how we can help. Obviously, I don't need to show you all these, but suffice to say that the mutations, largely 80% of the mutations are in this gene, the one gene, ATP1A3, reducing the activity of sodium potassium ATPase, as we heard this morning. And it's classically defined as a channelopathy, okay? So these are what the brain scans look like of these children. So these are CT scans of these children. So on the left is one child, on the right is the other child. So the classic thing that was emerging with mutations in Claudin-5 and AHC was calcification in the basal ganglia, okay? Calcification in the brainstem. And this is being seen in more than just these 2 children. I was mentioning earlier that I have been contacted by families in China who have the same mutation, their children have the same mutation, and they have the same clinical presentation of brain calcification. So brain calcification seems to be very, very common with these mutations. And this is the data that I'd mentioned about the arterial spin labeling. So to people not familiar with this technique, what it allows you to do is it allows you to simply label water. So you can use radiological mechanisms while you're lying in the MRI scanner, you can change the spin of water, okay? So as water enters the brain from the neck, you're able to measure the flow rate, okay? So how fast that water is moving. And essentially you're able to measure the flow of blood. This is one of that we had imaged using MRI at baseline, so at a time when she wasn't having a hemiplegic event. And this is during an active hemiplegic event, where what we're seeing here is a huge increase in red, a huge increase in flow on the right brain leading to the left-sided hemiplegia. And I do think this is the first evidence of arterial spin labeling as an MRI technique being used to show that there is actually a very heightened increase in blood flow on one side of the brain. These are the families. So mom and dad on both families are both, so these are de novo mutations, okay? Young girl here has the mutation. Young boy here has the mutation. His brother doesn't have the mutation. And what mutation is when I spoke about Claudin-5 and the structure as a transmembrane protein and the extracellular loops, the mutation is in one of those extracellular loops, okay? And it changes a glycine amino acid to an arginine amino acid, changing a neutral pH amino acid to a really, really heavily positively charged amino acid. And we had the hypothesis that what is happening here is that the channel that's formed by the tight junctions, it's turning a neutral channel into an anion selective channel. Okay, so it's turning the blood-brain barrier into a blood-brain channel, which that concept had never been described before. So we got very, very excited about this. What we were able to do was we were able to generate the genes, the human Claudin-5 gene and the mutated Claudin-5 gene. So we generated those in the lab and we looked at their levels, first of all. So remember, levels of Claudin-5 are really important. You decrease the levels, you get blood-brain barrier permeability. But there was no difference between the normal and the mutant Claudin-5. So this wasn't to do with decreases in Claudin-5 protein. What we term transwell inserts. Okay, these are save the cells in medium on the top part of them and on the bottom. So it's kind of like a cell culture based way of mimicking the blood-brain barrier. So you got the blood side on top and the brain side on the bottom. And what we were able to do is we could do these tracer experiments. So similar to what we do with the patients in the MRI scanner where we inject dyes, we can sprinkle dyes onto the apical part of the cells and watch how the dyes move across the monolayer. And you can clearly see in the presence of the mutation, the electrical resistance, so that the integrity of the junction is decreased, but you're also getting these massive increases in the flux of molecules from the blood side into the brain side. And while this is only a graph, this is the most important aspect of what we found. So what we've done here, growing the cells in these transwell inserts as well, on the top part of the cells, on the blood side, we've sprinkled in anions, okay, so chloride anions. And what you see here is in the presence of this G60R mutation in Claudin-5, you're seeing a massive flux of anions across the barrier because the barrier has been turned into an anion channel. Okay, so anions are flooding from the brain into the blood. And this is what we think is happening at the schematic level. So all the anions are here. They're normally excluded from getting into the brain because of the presence of the blood-brain barrier. But because of the anion selectivity now at this tight junction, the anions are flooding across the blood-brain barrier. So we published this piece in Brain just last year. As I said, the mutation is the first extracellular loop in Claudin-5. It's the first ever description of a mutation in the coding sequence of Claudin-5. Is it the second mutation or second or third mutation that's associated with AHC? As I said, it is incredibly similar to AHC. It likely has differences, but it's incredibly similar and it does fit all the diagnostic criteria of AHC as far as we know. Outside of this, it has implications for drug delivery in general and also the treatment of hemiplegia. So we're actively working on a treatment for this form of hemiplegia based on antibody therapy. So because it's in the blood, because the mutation and the protein product is in the blood vessel, we don't necessarily have to worry about the blood-brain barrier. We just need to get the therapy into the endothelial cell. So we're actively developing an antibody that will bind specifically to the mutant G60R Claudin-5 and not to the wild type, and therefore preserve barrier integrity. So we're very excited about this. The next steps for 2023 and beyond, this is an active project. So at the moment, we're making the humanized mouse. So we're making the G60R mouse. We finally have some embryos. So we thought it might have been embryonic lethal, which it shouldn't have been because it isn't in humans. We know it's not in humans. So we're making the mouse. As I said, we're creating therapeutic antibodies now to treat it. There are mechanisms by which, for ultra rare conditions, we can fast track this through the clinical development program to phase 1 treating or phase 2A as it will be. It also suggests that screening the blood-brain barrier in patients who have other forms of AHC, other genetic forms of AHC could be really worthwhile. So we're actively going to look into doing that in Ireland. Obviously, very, very few patients in Ireland is a big stumbling block for us, but I think this concept of screening the blood-brain barrier in patients, whether it's looking at the systemic profile, looking at biomarkers in the blood, or actually doing MRI scans with the dyes to look at barrier integrity. I do think that it's going to become very, very revealing for patients who are genetically diagnosed, but also for those patients who unfortunately don't have a diagnosis yet. Looking at the blood-brain barrier holistically in these patients will be very interesting. For anyone interested, I'm organizing a conference next year specifically on blood-brain barrier regulation across a whole range of disease from genetics to physiology. It's on in New Hampshire, beautiful New Hampshire in summer next year. And so you can take a look at that. I'll share these slides so everyone can get access to these slides. But this is where we're going to look at barriers of the CNS in general, genetics to physiology, and bench to bedside. And I do have a session dedicated to genetics within which AHC will be represented. So really excited about that. Finally, all this work, was not done by me. It's a village to do this work. So I'd really like to pay tribute to all the people, amazing scientists and clinicians that I work with in Dublin. And thank you very much for listening to me. Delighted to take questions.
I'm sorry, I was a little fast there. So how does it work that you have antibodies against the abnormal allele? It's just doing like a immunological ASO. What's the mechanism for that?
So we can generate antibodies specifically to the extracellular, because the mutation is in the extracellular loop of Claudin-5. We can generate antibodies that will target the wild type, the normal Claudin-5, but we can also target antibodies that will sequester the mutant Claudin-5. So from a therapeutic context, we can actually generate an antibody that will bind to the extracellular loop Claudin-5, but only in the presence of the G60R mutation. So it'll only bind to the arginine exclusive of the glycine. Yeah, so we have data in mice, that heterozygous mice that only have one Claudin-5 allele, are overtly normal. There's also a condition called 22q11 deletion syndrome. It's a neuropsychiatric condition that patients have microdeletions in chromosome 22q11, and that's where Claudin-5 is. And they're viable. Obviously, it's a plethora of comorbidities that they have because there's multiple genes deleted. But we do think that heterozygosity of Claudin-5 is enough to regulate these hemiplegic events.
I think you were first.
So for AHC, it's episodic. Is there research that the blood-brain barrier is only affected during an episode? Does it close up when they're normal? And does it just affect them on an episodic basis? Permanent, that's what I'm saying. And then also, is this just for hemiplegia or the other symptoms of AHC like brain development, dystonia? My girl has central apnea. Are those affected also by blood barrier? Have you looked into that as well, or is it just hemiplegia?
Time to digest that one.
Yeah. So the first question, the episodic aspect of it, yes, I do think it's episodic. I think that you're going to have the majority of blood-brain barrier disruption during the most intense clinical phase of what is happening, whether it's epilepsy or whether it's some other event. Absolutely. Now it resolves, and I do think what can happen sometimes is if the levels decrease too much, say for instance in the context of temporal lobe epilepsy, that I think that the threshold goes too low, that the Claudin-5 levels can't get back up to the levels they were before the seizure. And so with each event that happens, I think the Claudin-5 levels, the threshold will go down and down and down. We see that in traumatic brain injury, especially in repetitive mild traumatic brain injury. We do a lot of work on chronic traumatic encephalopathy, CTE, and we think that's the key hypothesis there, that repetitive head trauma is disrupting the blood-brain barrier. You're not leaving enough time to resolve, and it goes again, and the threshold just gets lower and lower. And your second question was related to specific symptoms. I don't have data for this, but I do believe that, again, there's a very, very strong correlation between the extent of barrier disruption and the extent of the clinical symptom that the clinicians will see.
Hi, I am very interested in your suggestions about a feasible way to kind of look at blood-brain barrier disruption in other AHC mutations that we already know about in ATP1A3. Is this something you can look at in mice? Is this something that imaging in patients would be helpful? What can we do as a community to kind of support research to look into that mechanism as a trigger for disease and as a possible route of intervention?
Yeah, that's a great question. Absolutely. So what we do, people talk about translational research, bench to bedside, but we do it the other way around. We generally start a lot of research programs with going to the bedside first and then proving it in mice. And we can do that in Ireland. It's an amazing way of doing it. So essentially looking at the clinical tools that we have available to actually do proper clinical research. So the question is absolutely. So we do contrast enhanced MRI on loads of projects in the lab at the moment, concussive brain injury projects, severe TBI, epilepsy. What we also do as well is we take images of the retina. So we use fluorescein as a dye to look at quantitatively at the integrity of the blood-retina barrier. These are all things that absolutely can be done. It's obviously going to be jurisdiction dependent. Obviously, the US, Canada will have different rules on the use of you know, what overtly are interventional dyes for clinical research, but certainly in Ireland, and I do think in Canada as well, that you can easily, I use easily in inverted commas, nothing is easy with clinical research, but absolutely, and something we could do quickly. Yeah, I mean, we circumvent these things, and you build that in if anesthesia is needed, and obviously it's not great, but if it's needed to actually get the clinical data, to get the results, it's all feasible. And we you know, certainly in Ireland, we have research MRI machines that have all of the anesthetic machinery that's MR compatible. The retinal imaging is something that we do. We do a lot of work on rare neurological diseases in Ireland, and patients just given mild sedation for retinal imaging. It's much easier because they kind of have to sit upright, and you try to catch them just before they get fully sedated, dilate their pupils and take detailed pictures of the retina at the back of the eye. So that's all possible. Certainly with the animals, animal modeling is straightforward. The problem there is, you know, sometimes you get spurious results in mice that aren't recapitulated in human, and vice versa. So I'm a really big believer in doing both, and sometimes doing the clinical research first rather than wasting money for bed and breakfast for a mouse.
I'm very interested about how specific the S100B is in terms of, because we have banked in our database in Mass General plasma and serum samples that were collected and handled very carefully. Wouldn't it be it'd be kind of interesting. I just don't know how specific that is. We did a study looking at RNA that showed upregulation that was quite variable, but inflammatory biomarkers from whole blood RNA, and we're just not sure how to pursue that. But could we send you plasma and serum samples from patients, and is that an assay that you could do as a quick look-see? That, you know, it's...
So the short answer is absolutely yes. Send me every sample you want and we'll look at them. I wouldn't ever look at one particular marker. So S100 beta has been around for years, and it's very useful, as I said, for telling whether the barrier is disrupted. It's very poor for prognosis. So to give you an example, there was a big study for patients who had out of hospital cardiac arrest and stroke. And obviously the S100 beta levels were very high in their blood, but it had no utility at all for the prognosis of those patients. Some had really, really high S100 and they were able to be discharged the following week. Others had marginally increased and they passed away 2 days later. So there are other markers. So what I would suggest doing is looking at up to 70 markers. We generally do Luminex arrays where we screen up to 70 markers, inflammatory markers, as well as blood-brain barrier biomarkers. And that's the best way to do it. So yep. Absolutely. Yeah, we can do that very quickly. Yeah.
So I was wondering if there's a blood test that would be amply available in the US for the blood-brain barrier disruption. And I had a second part to that question, which is how far are we from treatment based on this research, like from human treatment?
So there is no standalone blood test for looking at blood-brain barrier disruption. There are some jurisdictions in Sweden, for example, they use S100 beta routinely to assist with clinical diagnosis, but not in the US. So at the moment, no. There's a company called Banyan Bio based here in the US that have biomarkers available for traumatic brain injury in children. So making a decision as to whether the children should have a CT scan. So whether you should expose the child to radiation. And it's an exclusionary biomarker. So that's looking at GFAP and UCHL1. So Banyan Bio is the company. So you can look at that. But generally, I would recommend aligning yourself to research teams within universities to try to get access to these, large scale analyses. So don't rely on just one. Look at multiple factors. It's the best approach. And your second question was related to? Yeah. So absolutely, completely understand that the urgency for seeing work translated into human. The simple answer is I don't know. because there are so many moving parts to this that when we make this antibody and when we screen it in larger animals to prove safety first, that I don't know whether or not this antibody that we say is going to be therapeutic could actually be very, very detrimental to the brain. So very, very slow, incremental. And I absolutely hate putting, I know you want the number, the 5 years, 10 years, but it's just such a gray area.
Thank you. To the best of my knowledge, there's no ATP1A3 expression in any of the layers of the vasculature, possibly some innervation of it, but not in the actual layers. So there might be another clue though. Is there any connection in the children that have the claudin mutations to a relief of symptoms when they sleep? Because we know now that there are ion changes going on during the transition from sleep to waking.
That's a great question. And I did ask the treating physicians that exact same question. And they weren't able to do it because the children were all, during the phases of hemiplegia, they were all anesthetized. So they were all sedated during their active phases of hemiplegia. They were all hospitalized. So we actually we don't know whether they were sleeping or not because they were sedated. But absolutely, I do think that the blood-brain barrier disruption that we see with G60R likely is interacting with that ATP1A signaling axis as well. I've no doubt that it's feeding into an ATPA signaling axis. So I don't think this is just Claudin 5 and blood-brain barrier. There's something happening at the level of the neuron as well. Thank you.